So welcome back, everybody. Good morning, good afternoon, good evening, depending on where you are. We have some people on the panel for whom it is very definitely either very early in the morning or very late in the evening, and thanks to them for joining us. We've just had 5 days of meeting. We thought there were 3,000 people registered for the meeting. Somehow you have all logged in from about 4,500 different devices and IP addresses, which is fine. The meeting is free. And you've also logged in from 89 different countries. So we're humbled by the fact that you have joined us in this online meeting to discuss biodiversity genomics. So what we've organized now is a panel discussion with 6 people who I've called challengers, who are going to think about their experience of the meeting and their experience of biodiversity genomics and challenge us all with what they think we ought to be doing in the next year, in the next years, in order to complete our mad project of sequencing everything on Earth, in order to complete our mad project of understanding what it is we've done, and also to complete it with a proper sense of the world in which we live. So both in terms of sampling ecosystems ethically, but also working with our fellow human beings ethically with a sense of diversity and inclusion, and making sure that what we do includes all of our community. Okay, so that's the challenge to them. They've each got 3 minutes to put their challenge to all of us. And please put some of your questions to the challengers in the chat, because there is actually a delay between us talking to you here and it appearing on the screen in the real world. Some of your questions may arrive a little late, but don't worry, we keep a list of them and we'll make sure they get answered towards the end. So some of the questions may come from the rest of the panel. So I should say, as well as the challengers on this panel are also us 4 organizers of this meeting. And so we will also help by answering questions. So I'm going to start by asking Richard Durbin to issue his challenge to the community for the next few years.
Okay, well, thank you, Mark, and thank you all the organizers and everyone who contributed. It's been a really wonderful meeting. It's just an amazing range of science and also amazing progress, I feel, from. And I think the virtual aspect has, in fact, compared to previous, there have been previous iterations in some form of this meeting, but this has been a much more expansive event. So for me, I just want more of it. I mean, and that's really the top level challenge, is to scale. We have to be really ambitious about scaling, because it's hard to take genome projects. It's clear now that we can do this stuff, and it can change the way that we go about things, but we need to scale. And I've broken it down into 2 things, which I think are critical. There's 2 things that I want to say. First is coordination. It's great to see all this diversity blooming that we see, and we want to enable that and strengthen it and not impede it. But to scale, we also need to coordinate. And I think, EBP, who is a sponsor of this, is put itself in a place to do that. But there's a lot of work to do to coordinate. In particular, I think in the next year, we're going to really need to start coordinating species lists and sample lists. And otherwise, we're going to start treading on each other's toes. We already sequence many things many times, but that becomes embarrassing when it's at large scale. So that's urgent. The second thing is more complex, and you also referred to this. I think the legal and moral way forward actually is important. And I think actually this is harder. The data, we have to recognize the data from biodiversity genomics are valuable. I mean, and we're just going to carry on demonstrating more how valuable they are. And we who obtain the data have to engage with the moral questions about where the benefits should flow. And these are sort of political. People talk about legal and ethics, but I think there's a moral side to it. And if we ignore it, then things will not go the way that we want, I think. And we will be, in part, it would be wrong of us to point the finger and blame there. We need to engage, think about it. There are going to be some things, it's going to put constraints on what we do. We shouldn't just fight for what we have always done in the past. So we've got a lot to contribute to that, and we can influence it. But I think working out how to really engage with taking that forwards without being seen to control it, but it is a key issue for the coming few years. We're at a critical time. So I don't know where my 3 minutes are. I'll stop there, though.
That's good. Thank you, Richard. Anybody else from the panel want to comment on what Richard has just said?
Yeah, this is Eric. I'm going to second, even make a stronger argument that doing all these species, you don't want to have the EBP do them, or whatever projects out there do them over and over and over again. So we're going to need to get to the point, I would second that, of doing these genomes only once, meaning they have to be high quality, error-free, and telomere eventually.
Yeah, but I think it's important that each initiative doesn't think, oh, we're going to do all the sexy species. So how we're going to have to parcel it up. This was a problem in the Human Genome Project, and has been a problem in international genome projects, is stopping grabs for the most attractive things, and in a rational way, distributing the work.
For the moral part, yes, I agree.
Richard,
for the coordination part.
Yeah. We all know we're working on it. We know it's a serious problem, and it's already happening. It's not hypothetical anymore. And the issue is to really be able to collect these data from all the labs. The labs have to be willing to contribute. Even if you have a system, everyone needs to be willing to share the lists of species that they're working on. And we need a common format to do that. So that Fringy has kind of worked out a rudimentary way of doing this, a spreadsheet. We need to enhance that, but we need to encourage all the participants around the world to tell us what they're doing before they deposit things in GenBank. If it's already gets deposited, it's too late. We need early access. We need early sharing. And it is part of the coordination function, and we need to focus on it much more heavily this coming year. Thanks for the comment.
Yeah, I would agree. I'd give a plug for something we're doing in Tree of Life, which we're calling Genomes on a Tree, or GOAT, which is a database which not only, or a system, it's actually powered by Elasticsearch, which not only gives you an estimate of the genome size of any species you might care to ask, but is also going to be very easily a place where you can register an interest in the species. So you can publish, I am going to, or we are going to sequence this list of species, or I am sequencing this list of species, or I've nearly finished this species. And that will hopefully, this sort of system is really needed. I agree. One more comment before we move on.
This is Shen. So I think there's already some overlaps. But overlap is a good thing. I think theirs will be better if we have overlap. But sometimes gap is an issue. So overlap is not an issue. But overlap without conflict is important. So the coordination inside of EBP on an EBP framework is trying to solve this conflict. But we will have gaps. We will have like overlaps. I think it's a good thing to have overlap.
Good. So some overlaps, but not too many communication. I want to move on to our second challenger now, who is Montse Corminas from Barcelona. Montse, can you tell us what you would challenge us to do with your perceptions of the current state of the project?
Okay, thank you very much. First of all, I would like to thank the organizers for an incredible meeting and also for the invitation to be here today. And I would like to say a special thanks to Harris and Mark Blaxter, not only for the successful leadership in front of the EBP and the Darwin Tree of Life, but also because of their continued support and encouragement for our Catalan project. Our project is led by a group of people under the auspices of the Institute of Catalan Studies, which is an institution that works like an Academy of Science and Humanities. And if the project develops as expected, one challenge for us will be to better define which is the governance of this project in the near future. At the local level, I think that one of the main challenges that we face, like it's probably like everyone else, is funding. In our case, we have some financial support from the Institute and also from some local institutions that has allowed us to start some pilot projects. But we need to go one step further to serious launch the project. And this is what we have been putting all our efforts so far. And we are now discussing with representatives of both the public as well as the private sectors in order to warrant an investment for the implementation of the entire project. I think that we need to make the funding agencies understand the relevance of this project, and maybe having some background material that can be used across many different countries could be useful. Also, the European or more global level, one big challenge is coordination, funding including. This is what we're already discussing so far. So we need. I don't want to repeat that, but that's an important, very important issue. What I would like to say is that this meeting, the Biodiversity Genomics 2020, has actually helped to know about the different initiatives going on the continental Europe. This has contributed towards understanding a little bit of how to set this coordination. In this sense, I would like to emphasize that the European Genome Reference Atlas that was presented in the meeting by Claudio Scioffi is a great seed for the pan-European coordination and collaboration, and also hopefully to obtain substantial funding for the projects. I think it doesn't make sense that the UK has raised substantial funding for the Tree of Life, and at the same time, continental Europe remains paralyzed. And similarly, at the global level, I think one challenge is to find a way to fit or combine different strategies that do not have to be exclusive, but overlapping or even orthogonal. I mean with that I don't see this like as a inside national projects, at least within continental projects. There is obviously the orthogonality derived from the fact that some projects are taxon-based, some others are territory-based, but even within the territory-based, things are quite complex. In fact, if we think on biodiversity, the Mediterranean basin is one of the richest and most threatened biodiversity hotspots, and I think it includes 22 countries and 3 continents. And we, the Catalan project, feel that we have to work together and find common grounds with all these countries. So maybe one challenge will be perhaps to establish what could be the Mediterranean network inside this big confederated international network of networks that is always defined by hubs. Other challenge includes a wide range of social issues and communication, how to engage citizens at all levels, from field collectors to scientists and to politicians. In my opinion, this requires to be actively involved in education and wider presence in the media. I think that genomics and biodiversity should not be foreign or abstract words to the citizens. And finally, equity is also an important concept here. It's usually understood as empowering regions or countries so that they participate equally in the projects. I think it should be expected that not only species, but also methods and protocols to be contributed from participants. And I stop here, and thanks again for giving me the opportunity to share these thoughts with you.
Thank you, Montse. I'm going to put a question actually from the audience, which is to do with funding. So what about the idea of having high-level funding, independent of question or individual, and having then people involved who can then get involved without particular bias? So going for a global fund, which people can fit into for sequencing. That's from Emma Teeling. Sorry, Emma, that's...
But that's a question for me, or like a general question for everybody, I guess, right? Because it's
general for everybody. Sorry, Emma, that's paraphrasing.
I think that we should really do anything that is possible to get funding. I mean, funding is one of the most limited issues in this initiative, right? So it's like getting funding from global to small and small to big. I think anything should be possible, and it should be carried out. I mean, I think that's a very important issue.
Yeah, Mark, I think an answer to that would be sort of like, I guess Rich and others might know about this more, like the initial Human Genome Project, where you have different countries or different institutes participating in sequencing different chromosomes or different parts of the human genome. And maybe there's a more global funding mechanism that we can model off of for the EBP that would actually, benefit from that model.
I think that that's an important model, but again, this puts countries in the middle of everything, which is, I mean, we should take into account countries because funding, most of the funding comes from countries, and then the laws are based on countries. But still, I mean, species travel without borders, right? So I think that's one way to think, but not the only way. We think we have to find other ways after to organize them. I think that's why I meant this Mediterranean. I don't know exactly how to start doing that, but I think we have to look for institutes that cover that. So we have to think how to do, but I think it would be an important way also to move. Not base things only in countries. I think that
Quick
would
comment, quick response to Emma. Emma, thanks for your question. Indeed, that's where we started with the global funding. We really tried to do that. And what we found where there's resistance in two places to that. First, to just summarize one point is you got to walk before you can run. And you've got to convince people that you're real and that you can deliver genome. So there were questions at the beginning as to whether we could actually deliver on the project. And I think the last couple of years, especially the last year, has shown that indeed that we can do that. The other, from the point of view of philanthropies, who are really the only ones that can deliver on these kind of global projects, they like to see buy-in from the individual countries. They want to leverage their funds against state funding agencies and national funding agencies in a way of a match. So all these efforts now where individual countries and funding agencies within countries are contributing to national and regional projects are extremely important in our efforts in the coming years to get this more global funding that'll allow us, as Shun said, to fill the gaps and to proceed to phase 2 and to phase 3 of the project. I'm very optimistic about it because now we're walking, and I think soon we'll be running and in very nice Nike shoes.
Okay, I'm going to move on now to our third challenger. So our third challenger is Oliver Ryder from San Diego Zoo. Oliver, what is your challenge to the community?
Thank you. Congratulations to the organizers for producing a milestone conference that can be noted less for being inhibited by the pandemic than it will be for opening new doors for exchange, discussion, diversity, and inclusion. I'm energized, encouraged, and frankly thrilled with the progress made in many aspects of genomics present at this conference. I was enthralled by Richard Durbin's opening comments and Adam Phillippi's presentation describing the clever, elegant approaches used by the international team he led, resulting in significant improvement in the human genome assembly, gave substance to his statement that going forward, all sequenced genomes could be reference genomes. That statement reflects the undaunted spirit of enterprise that launched Genome 10K, the VGP, and the Earth Biogenome Project. We can anticipate wondrous new insights into life and its evolution as genome sequencing, assembly, and bioinformatics advances rapidly accumulate. For all of life, for human medicine, and for One Health, studies of organismal development requiring detailed and accurate genome annotation will need to be undertaken. This effort will require development of in vitro systems that can be built upon established and expanded resources of cryobanked viable cell cultures suitable for reprogramming to induce pluripotent stem cells. Thus, beyond sequencing genomes, we need to save genetic resources for conservation innovations, including genetic rescue. This will require expanded efforts in cryobanking viable reproductive or somatic cells. At both global and local levels, to achieve our goals in time to have our efforts fulfill their conservation destinies, the challenges include overcoming old historic patterns of colonialism, economic disparities, cultural and racial inequalities, and corruption. So societal change is an important component of achieving the goals and outlandish ambitions of Genome 10K, the VGP, and the Earth Biogenome Project. I'm uplifted by new initiatives led by young scientists who are devoted to addressing climate change, economic, social, gender, and racial inequalities, and conflict reduction that are likely to lead the way to more rapid accomplishment of societal goals, including caring for the Earth and its inhabitants, as, for example, envisioned in the Grow Columbia initiative. Our opportunities include no less than developing and using powerful new tools and technologies to contribute to conserving the legacy of life and the processes that produce it in new ways unimaginable a generation ago. Thanks.
Thank you, Ollie. Any immediate responses from the panel? If not, there's a question from the community about prioritizing species. So should we prioritize threatened species from low-income countries where funding is non-existent?
I think that's a very useful point of discussion. I think that we're losing, we can celebrate our progress, and we're losing biodiversity as we're talking, and the opportunity to describe it and bank it is being lost. And I think that those kind of priorities need to be established.
Yeah, Mark, that's my challenge I was going to talk about.
But we'll not discuss that any further. Carolyn? Good.
It's 4 a.m. here. I can't come up with a new idea if you take my idea.
You keep that out. That's Carolyn's idea. Any other comments?
Yeah, I just want to say, I said, Oliver has been saying for a number of years now that we need to bank cell lines. And this, of course, the genome itself, the annotation itself is expensive. Making cell lines would be expensive as well. But as we keep getting better and better, I want to second that's something that I would like to propose, that we try to add to our, recipe one day, or at least, things to do. Because that then allows people to go back to those same cells one day, but also do experimental studies and resurrect, whatever, if it's possible, species one day.
Oh,
Richard?
yeah, so I can see the value in that there's genetic resource there, but I want to, suggest that's a different project. And I think that partly, so I was involved in a project to make a bank of human genetic diversity and human IPS lines, made over 1,000 lines. Scaling that is an entirely different proposition to scaling sequencing, in my view, and that was all one species. So I think it's a really noble and valuable thing, but I think the goal of 1 million species cell lines, which I think is fully achievable, we can get 1 million genomes within the 10 to 15 year period if we organize ourselves right. I think that one would have to take a different approach for the cell lines and think through how to structure that, because I don't think that will scale in the same way, and I don't think the costs will match.
I don't disagree with you, Richard. I mean, it was purposefully heretical.
Yeah, that's
so I think it's valuable but different.
And as a botanist, which I'm not, I would point out that plants do this really nice thing of producing seeds which have some perdurance, especially if preserved properly. So some organisms work with us very carefully in that way, and seed banks are the possible way forward. I'm going to move us on, and our next challenger is Carolyn Hogg. Carolyn, I just want to say to you and all of the colleagues, and also Shunshu, all our colleagues in Australia, New Zealand, India and the East participating in this conference, thank you so much for staying up so late and or getting up so early. It's been great to have you at the meeting. I really enjoyed the two regional sessions that you had early morning my time. So thank you so much for doing that. But anyway, that said, what's your challenge to us, Carolyn?
Well, thanks. First of all, I'd like to thank everyone for inviting me today, but also all the fantastic presentations. I will say that I'm playing catch up every day rather than staying awake. I find by the time it gets to 3:30 in the morning, it's a little bit harsh to be concentrating. So my challenge really is quality. So I understand why we need these lovely chromosome length assemblies and to answer complex biological questions. And Eric really covered that so clearly in his plenary. But as a conservation biologist and someone who actually uses this information, quality for threatened species is never really going to get much better than where we're at now. And so my challenge is really out to all of those who work in the sequencing technologies and the algorithms about how we can better improve taking low quality, low grade DNA and being able to make a high quality genome for it. And so we've talked a lot about this week about genome quality and the multiple technologies that you need to make a chromosome length assembly to answer all your complex biological questions. But at this point in time, conservation questions don't really require that kind of quality genome. And the difficulty we're now finding ourselves in is that I would prefer to do a lot more genomes of lesser quality for more highly threatened species, which are dying at a rate of which we cannot possibly keep up with, even with all the everyone around the world doing all this global sequencing. And so what's happening for us now is when we go to a granting body or we send a paper out for review, or even it goes to a journal editor, the feedback we get is, but you're not doing chromosome length assembly. And I'm like, I don't need a chromosome length assembly in order to make decisions about what we should be doing for genetic rescue for this particular species. And so it's something that you need to think about when you're talking about the quality of genomes and what you're trying to do within the Earth Biogenome Project, is that you don't leave behind the people that actually need to use the information and apply it as a tool in their decision-making processes. So this conference is called the Biodiversity Genomics Conference, and it's not just about collecting all the genomes of all life on Earth. It's about how we use those genomes to protect all life on Earth. And so that comes back down to the equity issue. So if you look at it, there's 17 mega diverse countries in the world. Of those 17 countries, only 2 exist in the global north, and only one of them has discounted sequencing, and that's in the United States. And so it's not possible for the rest of the world with all these critically endangered species to be sending samples to Europe or China or the United States for sequencing, because we're not empowering the countries that actually have the most critical biodiversity on the planet. And so that's where Federica's talk was so fantastic about what they're doing with the Grow Columbia, about how do we empower those nations that don't have the funding, and how do we capacity build within those nations to help them progress their conservation decision-making and how to protect biodiversity. And Peter Dearden in our regional session raised the issue of cultural importance of all these animals and plants to many Indigenous communities around the world. So the cultural value to them not only lies in the species, but in their DNA and in their genetic code. And so being able to send samples overseas and sequence and store the data somewhere else is actually not a possibility for some countries. We can't get the permits to do it. Eric well and truly is versed in the bilby that we have not landed in Rockefeller, and in 12 months that we've been trying to get it out of Australia to the United States between the pandemic and CITES permitting. So it's something that we need to address. We can't just keep pushing. We need to do better and better quality genomes when the sample quality and the animals are not going to get any bigger or better quality. Plants make it easier to collect the sample, but then they have all those complex issues about how you sequence them. And so my challenge to the community is, when are we going to take a moment to think about how we are going to actually protect all the life we're proposing to sequence?
Thank you. Thank you very much.
Well,
I think I've terrified everyone.
Yes, no, I think one of the things we didn't realize is that we thought we can like I presented in my presentation, where we did some really interesting studies, like on the bird family tree and so forth, on the brain, with the more, I like to call them draft quality assemblies. But then we discovered when we got the higher quality ones, oh wow, there were things that that were important that we were missing. But I understand the point very well. And one thing I like to add to that is maybe think of the other side of the equation, is to try to improve the technology to more democratize it so that it becomes cheaper and doable in other countries. So samples don't have to be shipped, as well as for this community to try to work with the legal agencies to make things doable across countries, so we don't have to wait a year to try to get some approval. So I think that's a challenge I would add to your challenge, I should say, for the coming years.
Can I make a comment?
Yes, Marcela.
Yeah, go ahead, Marcela.
Yeah, so me as Brazilian, and during the diversity and inclusion talk, I think something that Thank God said really resonated with me, which is basically asking the communities in these very biodiverse places, what would they like to do first and to protect? So when I think about Brazil, basically, at the moment, the communities, they are living close to the very biodiverse forest. For example, they are usually very poor because the type of development is basically an industrial kind of way. So they want to deforest to create cities, to be able to have commerce and things like this, instead of having a bioeconomy. So I do think that the sequencing projects, they should bring that. They should bring an alternative, which is not just cattle and destroying the forest and sending land to, the cattle owners that want to put more. So the projects should take that into consideration, trying to twist the system around. So this is one thing that I've been thinking, because I always try to think about that and how we could solve that, but just for the sake of evolutionary biology, which we love, and I can do that for the rest of my life. So we won't be able to do it. We kind of need to bring a bioeconomy together with the science there.
Yeah. It's really important for stakeholder engagement to ask the people that are trying to preserve the planet at that interface what they actually need us to be working on in order for them to make smarter, more informed decisions around the management actions, their policy decisions, their funding decisions. The pandemic is sending the world into a global recession, and I can guarantee the first lot of money that is going to get pulled is conservation dollars. And I know that because I've seen it happen in the last, for the last 30 years. We've watched every time there's a global economic crisis, the first lot of funding that disappears is for conservation. And so that's a challenge that is coming pretty rapidly down the train tracks of those that work at the interface. And we really need help to work on species that require it.
Okay, I have a quick, the communities that you brought up is, that Carolyn brought up is very, very important. And for example, GIGA represents communities of communities, not just network of networks. So this represents, over 20 phyla that have no genome representation. And those are represented by people who are, been focused on these lineages for a long time. So I think we need this integration of these different communities at some point, because, the old adage is the whole is greater than the sum of the parts. So eventually we need to put these together and see how we can better integrate those, because it's, we're going to have to deal with all these the underrepresented lineages as well as communities, and the lineages live in, as mentioned, these biodiverse countries.
Okay, I want to hold that thought, Harris. I'm going to move on to Shunshu, who's going to challenge us with his view on where the community should go in the next year or years.
So in my head, we already met a lot of problems by doing projects under EBP. For example, when we do, although we have very ambitious goals to sequencing a lot of genomes. But when we're looking at the genome we have already finished about like more than 1,000, but more than half of them are vertebrate genomes. The reason is simple because they're easy. Compared to plants, they're much easier to sequencing. So the major issues when we met for the 10KP, the 10,000 plant genome projects we met is that the sample has always been the headache issues for the plant genomes, including the sample shipment across borders. And even we already get samples, the high quality genomes is really difficult. The high quality, high molecular weight DNA samples can be issues for some plant samples. And even we could get high molecular weight DNA, but the DNA is so limited. For example, when we sequencing some algae, it's really difficult to get enough DNA to starting the sequencing. So that's a issue. I think the issue could be, for sample issues, could be the species issues, or could be the technology issues, could be the policy issues when sample cross-border. So I think what we could do, the opportunities, I think, is the new technology, like a new sequencing or sample prep technology should further able to help us to solve these issues. So recently we did some works. For example, we're using a technology we call single tube LFR. Is one of the directions we think we can solve the problem, but still it's still too early. But at least in some cases, we already starting from the picogram DNA or directly from, for example, 100 cells to trying to solve the problem. But we're still on the way to solve the problem for the difficult or the species that underrepresent sometimes is because it's too difficult to get samples or to sequencing. So this is the issues, the challenge we already have at the moment. But in my mind, I think the challenge in the future, like years after years, could be the data mining, because we have so much genomes. Let's thinking about like we will have more than 1 million genomes available for the Earth Biogenome Project. How we do the genome comparisons over this 1 million genomes. I think we don't have the issues today because the genome we can do the comparison is so limited. But when we have like 1 million on hands, then the biological, theoretical, and computational challenge will be very significant. I think we don't have the algorithms on hand to tackle this problem. So I think that new algorithms to solve this genome comparison over 1 million genomes could be a new challenge, and this could be a new opportunity. I think because solve these new algorithms could be some new serials in algorithms, also new serials in the evolutionary serials. So I think these are opportunities for us. We are facing the challenge, but we also have opportunities in the future when we have 1 million genomes on hand for data mining.
Thank you, Xunshu. Any comments from the panel while I quickly check the questions from the audience? So one of the comments from the audience has been that that we're generating data at a breathtaking manner. I made a comment earlier that one of the conversations that my PhD was 2 genes worth of sequencing, and I have a PhD student who's just graduating with 50 genomes worth of sequencing. And so data archives, informatics, and resources, those are going to have to scale in ways which are n squared, not just n in terms of the number of genomes, but n squared for all those comparisons. And so that order of n squared is a real challenge to algorithms, and is a huge opportunity for data science. And that's something I think that lower and middle income countries really can excel in, where the data are produced elsewhere, but their algorithms, which use a data reduction process to just get at the important, the information, the signal in amongst the noise, is somewhere where bioinformaticians, informaticians, data scientists can really contribute. And that's a distributed activity. If the data are available centrally in the INSDC databases or other aggregative databases, then the process of doing that research can be truly international. Was there another hand up? Richard.
So absolutely, I think that computation should be distributed across the world, and the analysis and methods development, and that's really an opportunity. And education there is critical and enabling. I think it's not as bad as quadratic, and in fact, over the last 30 years, sequencing has become faster, has doubled at a faster rate than Moore's law consistently, and the algorithms have continued getting better. And it's because the more we sequence, the more it's like something else. Essentially, the calculations are linear or even sublinear, arguably, and we've lived off that with algorithmic improvement, and we're going to continue to need more of it. The total amounts of data in the EBP type work is actually, there are 1 million human genomes being sequenced right now. Is not the issue. It is investing in, it's a new type of, it's an information of a scale and type that hasn't had the attention that it's going to need. So there's enormous opportunities there in the data handling and analysis.
Yeah, I think the issues in the biodiversity genome is currently, because for the human genome, because for the health, for cancers, so a lot of people are focused on the algorithms on that part. But for the biodiversity, because genome to genomes, they're so different, and it's not from one species. So so that's why I think currently we already, when we working on for them 1,000 species from plants, then we will probably met the problem because the genome are so diverse to each other. But I think if we already have the genomes available on hand, and since people have the data to play with, the new algorithms will come out. I think it's the old new opportunities. This could be challenged, but I think it's new opportunities.
I see great hope for Oxford Nanopore technologies as it improves to democratize this exportation of capacities to these developing countries and around the world. So hopefully that that as that moves forward, that will be one way to expand, as well as having the data and the algorithms on the cloud. So that should be expansive as well, and that will allow other communities and countries to participate.
I'm going to move on to our last challenger, who's Marcela Uniano. Marcela, the floor is yours. Challenge us for the next year and the next decade.
Right, thank you. So I want to second all of the speakers today to congratulate Mark, Harris, Joe, Eric, Chloe, Sophie, and everybody for organizing this meeting, which was excellent. I want to challenge us in the perspective of the conference itself. So I think 2020 has been very difficult for everybody. But COVID or no COVID, this conference has really exceeded my expectations, but I think difficulty in pain is also give us ground to growth, right? So I think one thing that happens in everybody's mind during this conference is that we should have hybrid conferences in the future because two main reasons: it decreases our impact, our carbon footprint with let's say 20 or 30 less people flying around, which would be a very good thing, and inclusivity. So we could have developing countries, students and early career researchers joining, because especially in a moment of crisis that we are going to live in the next few years, it's impossible for them to pay for flights and to travel and pay conference fees in other countries. So this would be very good. But then, so of course, the person-to-person conference, we don't want to ever substitute completely, replace the contact that we have with other people. And then also, but if we are person-to-person, we don't want to be looking at our phones all the time because there will be online people. So I guess we do need to think on how to do that, I guess. So I've been to a conference that we had an app and everybody had a name and you could chat directly to the person that was in the conference. But if we don't want to do something fancy like that, we can go to Slack. So we can highly recommend everybody to engage on Slack. I think we should have also a list of everybody's names and email addresses, so people that would be online would be able to contact them with their new current email addresses. And I think during the talks we should have at least one question from the online audience, so we really engage them in that process. So it would be streamed live, and they would have a question. I think that would be very important. Right, and please no paper. If we go back to like person-to-person conference, we all have phones and computers. We don't need any papers or any gifts or anything. We work with biodiversity. Please, let's not have that, I would say. Right, I think we should keep talking about diversity and inclusion. As I said during the Diversity and Inclusion Forum, I think societal change, it depends massively on very big structural changes, but non-revolution in this world has happened just because some politician decided to. There is always a social movement together. And I think as we talk about diversity and inclusion, more it gets, like common ground by the whole society. So I think that's important. Last but not least, I think Oliver said that today too. I mean, a main thing about sequencing genomes, apart from conservation, is understanding evolutionary biology, which is amazing. And you have so many questions that you need answers for. And we are losing biodiversity in a very, very high speed. And for us to be able to understand how evolution happens and to be able to preserve species and everything, we do need to engage the whole society in the discussion. So I do think that we should have a showcase or like an open session for the public, or like have more journalists involved. I mean, scientists do already a lot. So not us, the scientists, need to do the communication there, but maybe we should have someone chairing and then invite some science communicators to do that, because we do need to engage society more in this endeavor, I would say. So that's it.
Thank you, Marcella. Any responses?
Yeah, I would say, Marcella, fantastic challenge. And for the EBP, and Harris can tell you for executive committee, as well as for the G10K council meeting, we'll be talking about this and take your comments to decide for the next year. And I want to say overall, my experience as being part of large genome scale efforts since like 2008, joining the G10K, B10K, we turned it to VGP, EBP, and now bringing us all together. This has been the most rapidly evolving conference I have ever seen. Each year it changes, and each year it's good. And each year it has been better, even with this online version. So I think if we follow that past spirit, I think with a lot of your advice, I think it will just get better and help us achieve the goals. With one of the biggest challenges being over the coming years, Harris, others just brought up, will be coordination. And I think this conference will help that coordination go forward.
I agree, and thank you, Marcella, for your comments. And I think that we may not yet really fully grasp the impact that this is going to have, and that how important it is to guide it in ways that contribute to, as interesting as the evolutionary biology is, as compelling as it is, as wondrous as it is. The way this is going to impact the planet is by changing people. And we need to make sure that that people like you could continue to give us that message.
Thank you. Thank you, everyone. I'm going to close with that statement and with Marcella's challenge to us all. Thank you very much, panelists, for participating in this. Thank you, everybody, for listening. I've got a few thank yous to say before the meeting closes, because this meeting happened online, and so you don't see the people running around in the background like you would do at a physical in-person meeting, making sure that everything works. You don't have the nice person jumping out of the AV box at the back of the room and running down to the front and fixing the microphones because we can't work them. So I've got a few people that I really must thank. So first of all the speakers. It was an odd thing to record our talks before giving them. And thank you to everybody who did so. That comes with a thanks on the side from the venue audio visual people. It makes it much easier for them to present the meeting, knowing that the talks are recorded and can be delivered. Thank you to the chairs for keeping people to time. Thank you for the chairs, especially also for raising to the challenges we gave you of putting these sessions together in quite short time periods. So thank you, all of those people. Behind the scenes, there's a bunch of people that nobody's seen at all who are the moderators. So for every session, we had a moderator who was the person who was sitting and making sure that the questions got through. So thank you to all of them. Again, a kind of thankless task because they're having to pay attention to the questions rather than the talk. So they missed out on a bit of a meeting so that we could have a fuller meeting ourselves. So thank you to all of them. Thank you to Venue Audio Visual, who kept us going even through a short outage during the Australia session, for which the internet in Britain apologises. It wasn't Venue AV's problem. Thank you especially from me, and I know from the four of us who organised this meeting, to the Tree of Life and the EBP administrative folks. So especially to Sophie and Chloe at the Tree of Life offices, except they're not in the offices, sitting in their kitchens at home, who have fielded all our questions and made sure that everything happens. So Chloe and Sophie, thank you very, very much. I'd like to thank PhiloPix. So if you haven't been to PhiloPix, it's a great place to go for a silhouette of your favorite organism. Those are the silhouettes I use to make the logo for the meeting. And if you haven't bought a t-shirt yet, do. And if you don't want to buy a t-shirt but still want the logo, do contact us at Tree of Life and we can send you the JPEG. It's all open source. Finally, I'd like to thank the sponsors. So we had 3 commercial sponsors for this meeting: Dovetail Genomics, Pacific Biosciences, and Oxford Nanopore. And their support was essential in making sure that we could deliver it to so many people. And so many people is the amazing thing. There are allegedly 3,000 of you registered. But as I say, there's many, many more people logging in. Maybe some of you are watching it on 2 screens at once. But thank you so much for joining us for this meeting. It's been a pleasure to be part of, and there are many, many talks that I really enjoyed, some amazing biology and some amazing work. And I think my hopes for the next year and the next decade are extremely buoyed by what we've achieved over the past 5 days. So thank you all very much. And with that, I think I close the meeting. Finally, I should thank my 3 co-organizers, Harris, Joe, and Eric, for making.
Can we also say a thanks to you, Mark?
And thanks to Mark. Yeah.
Yes.
You did an amazing
And
job.
the Sanger team.
There you go.
Sanger, thank
Yeah.
you.
Absolutely. So everybody go well, stay safe, have a good weekend, and return to your computers, desks, labs on Monday with renewed enthusiasm for this wonderful project we're all involved in. Thank you very much.